Welcome. Today I would like to present you the results I have obtained from analyzing the dataset from the laboratory of Dr. Cascardo. This project is about sleep deprivation in humans and how this affects sleep architecture. Before I show my results, let's go first through what is exactly the dataset. So the researchers collected sleep data from 4 subjects. EEG, EOG, and EMG data were recorded for each subject at baseline, that is without any treatment, so it's the control recording, and after a period of sleep deprivation that lasted 24 hours. The researchers then used the 3 types of recordings to determine the sleep stage throughout each recording. The sleep stages were classified as non-REM 1, non-REM 2, non-REM 3, non-REM 4, and REM. So for this project I have tried to answer to the following questions. How does the amount of time spent in each sleep stage change between the well-rested night and the sleep-deprived night? How much variability is there between time spent in each sleep stage between different subjects? And how does the amount of time spent in each sleep change throughout the night? So let's start with the first question, which is about the amount of time spent in each sleep stage. But before we even get to this, what happens to the total sleep time? Well, as we probably expected, the total sleep time increased. But then again, what happens to each sleep stage? Do they occupy the fraction of time throughout the whole duration of sleep? For example, the proportion of non-REM 1 to total sleep it decreased in the sleep deprived. In contrast, the fraction of non-REM 2 sleep remained stable. There was a trend for a decrease in the fraction of non-REM 3, but we would probably need a larger sample to be certain if there were any changes. Also, the fraction of non-REM 4 seems to be increased after sleep deprivation, whereas REM was unaffected. So our results show that sleep architecture was in fact affected by sleep deprivation. As I said earlier, we might need a larger sample to determine what happens to the fraction of non-REM 3. Why is that? Well, because the standard error of the means of these measurements is quite large, meaning that there's probably high variability between the subjects. Which gets us to the second question of this project. Before we proceed though, I would like to address a programming problem that came up with this data. The problem I have managed to address after all was to include error bars in the bar graphs, which was something we were not taught. So now let's go to the next question. How much variability is there between time spent in each sleep stage between different subjects? To address this issue, I have calculated the coefficient of variation, which is basically the ratio of the standard deviation to the mean. So it turns out that the coefficient of variation of the fraction of all sleep stages, except for non-REM 3, are increased after sleep deprivation, either a lot or slightly. In contrast, the coefficient for variation of the fraction of non-REM sleep decreased after sleep deprivation. Overall, most sleep stages showed a higher variability among subjects. But how is each sleep stage distributed throughout the whole sleep? This figure is a first approach to answer this question. So this figure shows the classification of sleep throughout the night for the control and deprived conditions of a subject. And this is the point where another problem appeared. Remember that the total sleep was larger after sleep deprivation. So if we plot one over the other using the actual time on the x-axis, we can't really compare the two conditions. To overcome this problem, I have normalized the time spent in each sleep stage to the total time. So this graph shows the sleep states that the subject was at each time fraction of the total sleep. You can tell, for example, here that at half the time of the sleep, the control control conditions was in REM sleep, whereas when deprived at half time it was at non-REM 2 or 1. Nevertheless, this approach still does not help us a lot, because although we kind of get an idea of what happens, we can't tell exactly how different sleep stages are distributed throughout the whole sleep. So here's another approach that I think it's a bit more clear. So this graph shows the fraction of the total non-REM 1 exhibited through the recording versus the fraction of the total sleep time. For example, in non-REM 3, as we see here, or non-REM 4, you can see that the red one, which is the deprived, reaches close to its maximum here and here, a lot earlier than the control. So this gives you a clearer picture of how each sleep stage is distributed over the whole recording. And you can see that the deeper phases of sleep, non-REM 3 and non-REM 4, reach their maximum sooner in the deprived than the control. Of course, all this is only for one subject, but all subjects showed similar pattern. So, in conclusion, we've shown that sleep deprivation changes sleep architecture in different ways. First of all, total sleep is prolonged. Second, the variability of the fraction of each sleep stage between subjects is increased after sleep deprivation. And third, sleep deprivation seems to cause higher prevalence on the early phase of sleep. Further work should probably attempt to link these results to changes in behavioral performance such as in memory or attentional tasks. Thank you very much.